Okay, so tutorial number 3. Alright, so we've gone through differences between plant cells and animal cells. We've gone through different organelles and what they're responsible for. We've done active transport and passive transport, and we've looked at how molecules and things get in and out of the cell. So now I'm going to move on to surface area to volume ratios. Okay, so let's just have a look at this first. Surface area, basically that means how much is exposed to kind of the outer world. So the surface area, say, of this cube would be all of the faces of the cube added together, and that number will be the total surface area of this cube. Okay, the volume, when we talk about volume, that's basically how much stuff is in there, how like what the, is there 1 litre, 2 litres, 3 litres, so basically the volume, what is in there. So let's say this here weighs maybe, 1 litre, okay, and the surface area is, let's say, 1 metre, okay, so your surface area to volume ratio will be 1 metre to 1 litre. Okay, so basically if we've got this cube, okay, so I was talking about in class something that we called the Weet-Bix theory. I really just made that up, so don't say that in your assignments. Okay, so basically what this is I just kind of think about it as I've got a couple of Weet-Bix. I have Weet-Bix on my breakfast. That's the first thing that came to my head. Okay, so I have 2 Weet-Bix, say. Okay, and then I pour my milk on. Okay, and basically the milk soaks around, the milk will soak around all of the outside bits of the Weet-Bix, but often what I find is that the inside of the Weet-Bix is still kind of dry. Alright, so you need to keep adding more and more milk to try and get that little inside bit dry. Okay, to get that little inside bit wet. Now, if I was to, instead of having my Weet-Bix like that, if I was to break it all up, okay, and kind of have like a Weet-Bix, like broken up bits, okay, then I pour my milk on, what's going to happen is all these little bits are going to soak up all the milk, and I'll end up with a really squishy, kind of disgusting looking thing, okay, but no dry bits. Okay, so what's the difference between these two breakfasts? Okay, so this one has been broken all up, so this has a higher surface area. Okay, use SA as surface area. Okay, and this one has a lower surface area. So we've still got 2 Weet-Bix. We've still got the same amount of Weet-Bix in here. Here I've just crushed up the Weet-Bix. So we've got 2 Weet-Bix, 2 Weet-Bix. So the volume is the same. Okay. What's changed is the surface area. Okay. So this one has a higher, we say that it has a higher surface area to volume ratio. Okay, and when things have a higher surface area to volume ratio, they're going to be much better at absorbing. Okay, so that's absorbed all the milk, whereas that one, it hasn't. It hasn't. These bits in the middle are still kind of dry. Okay, so why is this important? Okay, number 1, because cells are only, so let's say here's a cell, here's our little nucleus. Okay, so cells are really small. Okay, so why don't we have huge cells roaming around everywhere? Why are cells so small? Okay, and the reason for this is because these things are unicellular organisms. Okay, let's pretend we have 1 cell. Okay, so this is our cell. Why are cells not bigger? Well, cells rely on diffusion and on osmosis. So the way that they get their gases, so oxygen, carbon dioxide, water, okay, so the way they get these in and out is through what we call their semi-permeable membrane. We've talked about that. Okay, and this allows molecules to come in and out, okay, through something that we call diffusion. Alrighty, so diffusion can only really happen over certain distances. Okay, so if the distance is too far, say the cell is this big instead, what might happen is the gases and the oxygen and things like that, they can't get to the middle of the cell as easily. Okay, so in here it's quite a short distance. They can get to the cell's centre really easily, whereas here they've got a much longer way to travel, and essentially the places in the middle aren't getting all the nutrients they need. So yeah, that's basically why they can't be any bigger than they are. This also happens for unicellular organisms. Okay, that's a reason why we don't have ginormous bacteria organisms. Why we don't have ginormous bacteria, you know, kind of floating around everywhere. That's because they are unicellular, so they're only one cell. So the same thing applies to them. If they get too big, this centre bit here, it's taking too long for nutrients to get to the middle of the cell, just like the dry Weebics. Okay, so basically that's why things are so small, because the middle bit will basically essentially die. Okay, so now how do we increase surface area to volume ratio? So we had a look at this thing here. Basically, the way that you increase surface area to volume ratio is to to break it up into smaller little bits. Okay, so smaller little bits will have greater surface area compared to the bigger bits. Okay, so for example, with this little cube here, if I chopped that cube in half, okay, I'm immediately exposing another edge on this side of this one and another edge on this side of this one. Okay, so you end up with two. Oh, my drawing is not great. Okay, so you end up with two that look like that, and this is that one there, and then on this side behind there would be totally new for molecules to come in. Okay, so breaking it up basically, so I mean if you think about a square like this, okay, and then if I do the same volume but stretch it out, okay, that's going to have a greater surface area. There's much more stuff here. Okay, so that's another way of increasing surface area, making things really skinny. Okay, so basically that is that, surface area to volume ratio. Alrighty. Okay, so what have we got now? Okay, so now important bits. Photosynthesis and respiration. We will start with photosynthesis. Okay, we'll do photosynthesis and then we'll have a break. Okay, so basically photosynthesis, we're going to go with our definitions again. Photosynthesis is the process by which light energy is taken and converted into sugar. Okay, and then this sugar is going to come in when we talk about respiration. Okay, so things that photosynthesize are called autotrophs. These are things like plants, algae that photosynthesize, bacteria. Some bacteria photosynthesize. Okay, but basically what we're going to be talking about is mostly just plants. Okay, so photosynthesis is two different parts. So you got your what you call the light cycle and the Kelvin cycle. You can think of it as the dark cycle, but technically it's slightly incorrect because the dark cycle actually happens during the light as well. It just doesn't happen as much as the light cycle happens. So either one is generally fine. It's better practice to use Kelvin cycle rather than dark cycle. Okay, so basically we had our little diagrams in our book, so if you can find that, that'll be cool. Otherwise I'm going to draw a picture, which as you know, not great. Okay, so light cycle. So this is where you got the sun, Rays come down. Here is our plant. Okay, so it's obviously not going to go onto the petals, alright, it's going to go onto the leaves, the green bits. Okay, so basically the light hits the cells in the leaves that are going to pick up the light, so that'll be the chlorophyll molecules inside of each of the cells. So we've got our cell here, and oh, that's a terrible drawing. Yeah, okay, so we got our cell here, and we got all our little chloroplasts, so we saw that in our microscopes topic. Okay, and basically it's going to go down, it's going to hit one of these chloroplasts, and that's where the light is captured. Okay, so if I just draw a little chloroplast here. Okay, here's my chloroplast. And I'm drawing some stacks that look kind of like pancakes. These are called grana. Okay, and each one of those little pancakes is called a thylakoid. And it is on the thylakoid that the light hits. Okay, so that's the first stage. Light comes in and it bounces onto one of these thylakoid membranes. Okay, so I'm going to write that in, thylakoid membrane. Here, I'll start drawing. Okay, here's my chloroplast. Okay, so basically the light comes in and bounces onto these thylakoid membranes. And what it does is it excites the electrons that are chilling in the membrane. Okay, so it basically gives them a spark of energy and they start kind of freaking out. Alright, so they basically start, start jumping around, alright? So they start jumping around on the membrane, and that causes a whole bunch of energy to be transferred. And so a whole bunch of this energy is basically, so we go along very similar to the electron transport chain. Okay, so electrons are bouncing around. Okay, and what happens here is it charges up ATP, so ADP to ATP. Okay, and it also charges up NAD to NADH. Okay, during this process, water comes in, okay, H2O, and basically the water is kind of travels through the membranes, and water is split. So if you think about H2O, if you split that up, you get hydrogen and oxygen gas, yeah? So it's gas and that's, okay, so that's not totally balanced, so don't worry about that. Okay, so oxygen is released. Okay, so that's where the oxygen is released. Plants release oxygen. Okay, and the hydrogen basically jumps on here to the NAD. So that means NADH, having the H there means that it has electrons. Okay, and the hydrogens come out and they balance that. Okay, so that's the light cycle. So basically, so far we have, so we've got, here is our photosynthesis equation. We got H2O plus carbon dioxide in the presence of light and nutrients. We get C6H12O6 glucose, sugar, okay, plus oxygen released as a byproduct. Okay, so here we have our first byproduct that's come out of the light cycle. Okay, so that's where that oxygen has come from. And these hydrogens have been bounced onto the electron transport, onto the electron carrier, NADH. Okay, so that's our first cycle. Okay, second cycle is called the Kelvin cycle. And basically here is just a whole bunch of different reactions, and it's like a cycle, very similar to the Krebs cycle. Okay, but this cycle can only work with the help of the ATP and the NADH that has been produced in the light cycle. So ATP is needed here and NADH. Okay, these are used up during the cycle, and then the ADP, so the phosphate is knocked off, electrons have been taken, and these go back to the light cycle to be recharged up again. Okay, so basically a Calvin cycle, what you do is you're taking a bunch of molecules and basically you're changing them. These arrows meaning that you're changing them. Remember, you've always got enzymes that are associated with these. Okay, and you're bringing it down, you're making some modifications, and basically the end product you'll end up with is some kind of sugar. So the plant could store that as starch, so like potatoes. It could use the sugar for itself, so that sugar will come back into respiration. So we'll talk about that next. Okay, and here is where in the cycle, right here, right at the start, here is where you've got your carbon dioxide coming in. So here is our CO2, and that CO2 binds with this to form, what is that molecule? Anyways, this bonds with basically this chemical plus CO2 goes into this, and then it gets a whole bunch of modifications, and it ends up as a sugar, which is the important bit. Alright, so basically then you end up with your sugar, and then that'll lead on to respiration, or in the case of animals, the animals will eat the leaves and things, or eat the potatoes that contain the starch, contain the sugar, and then that'll lead into respiration as well. So all energy ultimately has come from plants through photosynthesis. Okay, so just one more thing before we go away from the chloroplast. Okay, so if this is a chloroplast, here these were supposed to be my light rays. Okay, light's coming into the thylakoid. So this Calvin cycle here, that's happening in what we call the stroma. And that's kind of the liquidy, stuff that all the thylakoid, all the thylakoids are kind of sitting in. So the stroma is where the Calvin cycle happens, and the light cycle happens on the thylakoid membranes. So you need to be able to label this. I would draw that if you get a question like this in the exam. I would draw the chloroplast. This is the chloroplast, and I would label it. I would draw all these things in here, and I would identify the fact that the light cycle happens here, and the dark cycle happens there. Cool. Okay, so respiration next.